The first question that we discussed today is digestion and absorption of proteins. Proteins, they are made up of amino acid, and this amino acid, they are connected together by peptide bonds. So hydrolysis of this specific peptide bonds, it is done by a group of enzyme known as peptidases. Peptidases, they belong to hydrolases, that is class 3 of enzymes. All these peptidases, they are secreted as inactive zymogen and later they are converted into their active form. So peptidases, they are classified into two types, endopeptidase and exopeptidase. Endopeptidase means the one which is acting on the peptide bond inside the protein molecule. And exopeptidases are those which are acting on the peptide bonds located at the ends of the polypeptide chain. And exopeptidase, it is again classified into carboxypeptidase, which is acting on the carboxy terminal, and aminopeptidase, the one which is acting on the amino terminal end of the chain. Hydrochloric acid, which is present in the stomach, makes the pH optimum for the action of pepsin, and it converts pepsinogen into pepsin. Then renin, it is also known as chymosin. It is active only in infant and it is responsible for curdling of milk. So it will convert milk protein casein into paracasein. And pepsin, which is secreted by chief cell, it is secreted as pepsinogen. HCl will convert this pepsinogen into pepsin. So by the action of the enzymes present in stomach, proteins are converted into proteoses and peptones. Next is the digestion in pancreas. Pancreatic juice contain 4 enzymes: trypsin, chymotrypsin, elastase and carboxypeptidase. All these 4 enzymes, they are secreted as zymogens. Trypsinogen, it is activated by enterokinase, which is present on the intestinal microvillus membrane. Once it is activated, it will activate other enzyme molecules also. The function of trypsin is to catalyze hydrolysis of the bonds formed by the carboxyl group of arginine and lysine. Trypsin will convert chymotrypsinogen into chymotrypsin. In this trypsin and chymotrypsin, they will degrade the protein into small peptides, and these are further hydrolyzed into dipeptide and tripeptide by carboxypeptidase. So the procarboxypeptidase, it is converted to active carboxypeptidase by trypsin. So the products of pancreatic digestion include oligopeptide and amino acid. The complete digestion of small peptides to amino acid, it occurs by the enzymes in the intestinal juice. So the luminal surface of the epithelial cell contain the following enzyme, that is leucine aminopeptidase, which release the N-terminal basic amino acid and glycine. Then proline aminopeptidase, which remove proline from the end of polypeptide. Then dipeptide and tripeptide, dipeptidases and tripeptidase, which will bring about the complete digestion of protein. The absorption of amino acid, it occurs mainly in the small intestine. It is an energy requiring process, and there are transport systems which are carrier mediated. And there are different carriers for neutral amino acid, basic amino acid, amino acid and glycine, and acidic amino acid. The absorption of neutral amino acid in intestine, kidney, tubule and brain is by Miester cycle, and here the main role is done by glutathione. So glutathione, it reacts with the amino acid to form gamma-glutamyl amino acid, and this reaction, it is catalyzed by gamma-glutamyl transferase. So the glutamyl amino acid, it is then cleaved to give free amino acid. So the net result is the transfer of amino acid across the membrane. And the transport of one molecule of amino acid and regeneration of glutathione requires 3 molecules of ATP. Second question is proteolytic enzyme. So the dietary protein, they are digested by a group of enzymes known as proteolytic enzyme. These proteolytic enzymes are of two types, endopeptidase and exopeptidase. And all these proteolytic enzymes, they belong to hydrolases, that is class 3 of enzyme. They are secreted as inactive zymogen, and they are later converted to their active form in the intestinal lumen. And this is to prevent the autodigestion. As we said, it is of two types. The proteolytic enzymes are of two types, endopeptidase and exopeptidase. Entopeptidases are those which are acting on the peptide bond present inside the protein. It includes pepsin, trypsin, chymotrypsin, and elastase. Exopeptidases, they act on the peptide bond at the ends of the polypeptide chain. Carboxypeptidases, they act on the peptide bond at the carboxy terminal, and aminopeptidase, they act on the peptide bond at the amino terminal end of the chain. Third question is, what are exopeptidases, and mention example. We have already discussed exopeptidases. They act only on the peptide bond located at the ends of the polypeptide chain. It includes carboxypeptidases, which act on the peptide bond at the carboxy terminal, and aminopeptidase, which act on the peptide bond at the amino terminal end. Next question, name the enzyme defect in maple syrup urine disease. Answer is branch chain keto acid decarboxylase. Fifth question is decarboxylation product of histidine. It is histamine. Then list the compounds formed from arginine. So the compounds formed from arginine include nitric oxide, creatine, and urea. Importance of glutamine. The third and ninth nitrogen atom of purine is derived from glutamine. And the third nitrogen of pyrimidine is from glutamine. Then glutamine, it is the source of amino group of guanine and cytosine. It is also a conjugating agent, and it donates the amino group of amino sugars and amide group of nicotinamide. Next question is GABA. So glutamic acid on decarboxylation gives GABA, that is gamma-amino butyric acid. It is an inhibitory neurotransmitter because it opens the chloride channel present in the postsynaptic membrane in central nervous system. Then the biological function of glutathione. Glutathione, it is a tripeptide formed from glutamic acid, cysteine, and glycine. So it has got a role in the transport of amino acid, which we have already discussed. That is the Miester cycle. Its role in Miester cycle. So you have to write about the role of glutathione in amino acid transport. Then glutathione, it is present in RBC. This is used for the inactivation of free radical, which is formed inside the RBC. Then glutathione, it is necessary for the reduction of Met-hemoglobin to normal hemoglobin, because Met-hemoglobin, it is unavailable for oxygen transport. Then glutathione, it helps to detoxify several compounds. Then glutathione keeps the enzymes in reduced active state. So these are the functions of glutathione. Next question is, name the polyamines and mention its function. So polyamines are putrescine, spermidine, and spermine. They are synthesized from ornithine with the help of methionine. The functions of polyamines include, they are required for the protein biosynthesis. Several roles are suggested for polyamines in cell proliferation, synthesis of DNA and RNA. Then polyamines concentration is increased in cancer tissue. And polyamines are growth factors in the cell culture systems. Formation and functions of nitric oxide. Nitric oxide, it is formed from arginine by the enzyme nitric oxide synthase. It contains heme, FAD, FMN, NADPH, and tetrahydrobiopterin. The functions of nitric oxide include, it relaxes the smooth muscle, then it prevents the aggression of platelets, then it functions as a neurotransmitter in the brain, and it also mediates bactericidal action of macrophages. That is, this nitric oxide, it is a vasodilator. The normal blood pressure is maintained by nitric oxide, and it also causes the cerebral, coronary, renal, and muscle arteries to dilate. Then nitric oxide, as we said, it inhibits the adhesion of platelet and so depresses the platelet function. It is a non-adrenergic and non-cholinergic neurotransmitter, especially in the gastrointestinal tract and urogenital tract. So it really relaxes the smooth muscle and leads to reduced gastrointestinal motility. Then you have to write about its role in disease and treatment. For example, inhalation of nitric oxide, it is useful in the treatment of pulmonary hypertension,